Okay, let's go ahead and get started. So today is second to last lecture. So we're going to continue our discussion of MRI. And then if I forget, remind me at the end of lecture today, we'll talk a little bit about project logistics and any questions you may have. So before I begin, are there any questions from last time or any general questions? Okay. So hopefully everyone had a good Thanksgiving. And so today we're going to finish our discussion of functional MRI and then talk about diffusion weighted imaging and diffusion tensor imaging, which are very sort of widely used applications of MRI. But before I do that, I did want to address some of the things that came up on the pre-lecture quiz. So reminder, this is a picture or a screenshot of what a console looks like for a GE MRI system. And one of the things we talked about before was when we set the parameters, the user typically, one of the key parameters they have to set is the field of view, so how large the object is. And then we talked about how field of view relates to our delta K sampling, right? The other thing we talked about was the resolution, and that goes into sort of how wide we go in K space. But you notice here on this screen, there's no place for resolution. And typically, a lot of consoles don't have resolution, but you infer the resolution from the matrix size here. Okay. So in this case, delta X would be field of view over 256. Okay. So that'd be whatever that 24 centimeters over 256 is. And delta Y in this case, we would look at the field of view in the phase is 100% of the readout phase. So that would still be 24. And notice here, we only have 128 lines in the phase encode direction or the Y direction. So the delta Y in this case is bigger than delta X. All right. Oh, the color scheme is different on that. That's interesting. Looks blue on my iPad. That's cool. All right. So anyways, so one of the things that questions I came up was with aliasing. Let's see if no, these pictures look okay. All right. Oh no, it does change. It totally flips the colors. That is so weird. Okay. Um, so the Gibbs artifact, one of the questions on the pre-lecture quiz was Gibbs artifact. So remember again, Gibbs artifact is this ringing that we see here, right? And so the question is, what causes that ringing? And in this case, you notice I only acquired 128 lines in K-space in the phase encode direction. So that means in terms of K-space, I only went out, for example, maybe this far in K-space, okay? So along the KY direction. And if you think about it, the resolution here, the delta Y here is, twice the delta X, right? Because if I have the same field of view, I only have 128 lines going in the Y direction and 250 lines going in the, well, let's call this the Y direction, sorry. And this is the X direction, just for this case, okay? So in this case, what I have to do is I have to further go out in K space. So I get 256 lines in the phase encode direction. And if I do that, then I get rid of I can reduce that Gibbs artifact. So that's one approach that you can take, that technicians will typically take. And in this case, then, since I have 256 lines in both directions, then in this case, my resolution is the same in both directions, right? So that's what that question was getting at in terms of Gibbs artifact. The other thing that people sort of seem maybe need to review is the idea of what happens as I increase B0, right? Why does the T1 increase? Okay. And the answer is it increases because remember the Larmor frequency, is proportional to B naught. Okay. So the precession frequency is proportional to B naught. Remember T1 acts best when the tumbling rate is close to the Larmor frequency. And so if I were to move the Larmor frequency, so this was my initial Larmor frequency. If I move to a higher frequency here, okay, so this is a higher F naught, let's say, then the idea is there's just not as many spins available there that have the right tumbling rate. So that's why the T1 gets longer. All right. So that's sort of an interaction between what you're doing for the experiment and just the nature of water and how things tumble around. So it really depends on the local environment. All right. So it is true that as you increase B naught, the polarization M does increase. Okay. So you do get more magnetization, but that's not the primary effect for the T1. Is there a question there? No, just all right. Okay, so if there's no more questions, let's move on, and we're going to sort of review a little bit about what we did last time with fMRI. So remember, we're looking and understanding brain function. We talked about the bold signal change, where what we're looking at is R2 star or T2 star decay. Okay, so basically how things decay, and we typically acquire at some echo time. Okay, and we're looking at the signal here. And then what we notice empirically is that when someone does some mental activity, then that signal tends to go up. Okay. And so therefore, if we're acquiring the same echo time, this means this delta R2 star must have been gone down. Okay. So R2 star A must be less than R2 star B. So the relaxation rate must have gone down with activation. And we can write down, typically the bold signal change is written like this. It's a sort of relative bold signal change. So it's the activation signal minus the baseline divided by the baseline. So it's the percent increase. And if you do a first order Taylor series approximation, you can sort of show that it's proportional to the echo time times the change in delta R2 star. And we just said that, so this is less than 0. And we just said that this is also less than 0. So overall, we're going to get a positive increase in bold. Okay, that's not always the case, but it's a good rule of thumb that in general, when the brain is more active, you have generally an increase in the bold signal. Okay, and so reminder of what we talked about is the main driver of R2 star is how much deoxyhemoglobin you have there. So deoxyhemoglobin is essentially the hemoglobin molecule with the oxygen stripped off. All right. And so the idea is when you're in the lungs, so you're in the lungs here, and so you basically have oxyhemoglobin, right? So basically the hemoglobin molecules are carrying the oxygen atoms into the tissue, and as they offload the oxygen, then now we've gone into the brain, and now they're started offloading those oxygen atoms. So the oxygen, the O2, goes into the brain, and so now we have these iron atoms that are exposed. All right, and so these are what causes the dephasing in the magnetic field. It basically perturbs the magnetic field around it, and that leads to more dephasing. So if we have a little bit of deoxyhemoglobin, there's some deoxyhemoglobin and there's some dephasing. And so that reduces our signal amplitude. And the more deoxyhemoglobin we have, the more dephasing there is. And so that's going to decrease our signal amplitude. Okay. This also leads to a higher R2 star value. Okay, and so then we talked about what really causes, what causes the how much deoxyhemoglobin you're going to have. And it's typically going to be mostly in the veins, although there is some, now there's thought that there might be some effects even in the arterioles, that you're already losing some oxygen even before you get to the capillaries. But it turns out it's a trade-off between two factors. One is the cerebral metabolic rate of oxygen. This is how much oxygen you're using up, right? And human brains are really, really greedy for energy, right? So for example, on average, the human brain weighs it's like 2 to 3 to 4% of your body weight, right? But it uses like 20 to 25% of your energy, right? So from an evolutionary point of view, it must have been really important because it's a small thing, but it's taking up most of the energy budget, right? So oxygen metabolism is really, really important, right? And all of you know that because how long can you hold your breath, right? You can't really hold your, I mean, unless you train, most of you, it'd be hard to, you would pass out if I took away your oxygen, right? So limited supply of oxygen. So oxygen is really important for keeping your brain running. And so the more you use up the oxygen, then you would expect deoxyhemoglobin to go up, right? But the brain is also says, well, I know you're going to use up oxygen, so I better deliver oxygen. And that's what's called cerebral blood flow. So you're constantly delivering oxygen. Typically, on average, if I wait 1 second and I look at any given part of your brain, 1% of the volume of that brain will be new blood. All right. So that's how much blood you're getting. Basically, every second so in 100 seconds, basically, you'll have replaced all the oxygen. Okay. So that sort of makes sense, right? Because, on average, I mean, thinking about holding your breath for 100 seconds, unless you train for it, that's probably going to be the limit of what you think you can do. All right. Okay. So cerebral blood flow tends to drive down deoxyhemoglobin, and oxygen metabolism tends to drive up deoxyhemoglobin. So those are the two competing factors. And we know from observation that in general, with activity, we expect DHB to go down. Okay. So that's the observation because the bolt signal goes up. So therefore, the amount of deoxyhemoglobin must have gone down. And therefore, in this competition between blood flow and metabolism, somehow blood flow is winning out. All right. And we talked before, it's anywhere from like a 2-to-1 to a 4-to-1 ratio of CBF, the change in CBF to the change in metabolism. All right. So as we talked about, this means it sort of makes sense if you're going to design a system that has to survive and has to outrun predators. And, you don't want to if you just sort of matched your oxygen level with your metabolism, you're right at the border, right? And so basically you risk the chance of, if you really need more metabolism, you're going to run out of blood flow, and maybe your thinking ability decreases and everything like that, right? So from an evolutionary point of view, it sort of makes sense that you would always want to have more blood flow than you think you need, right? So that's the bottom line on the bold mechanism. This is a nice graphic so this is really interesting because I just realized, I'm not sure this happens every time, but the colors are totally flipped. Does it happen all the time?
I noticed that you mentioned red when it's blue.
Okay.
Previously, in
Yeah.
other classes.
Yeah.
So maybe
That's really funny. It took me to the end of the pins. Oh, okay. Yeah. Let me try.
the icons.
Every lecture? I don't think it's every lecture. Yeah,
Okay.
maybe.
Oh, okay. I don't want to sort of. All right. Well, let's just I'll play with it later. Okay. So anyways, blue is red and red is blue. Okay. So you have to use your brain more now. Okay. So
DHB goes up.
the what?
DHB goes up. No,
Yeah, I think the arrows are okay. It's just the colors are like swapped, blue and red. I mean, it's really weird because I mean, like full swap of blue and red is like, how does that happen with RGB? The pins are, yeah, but then, I mean,
Connect to the screen. It's doing the blue.
oh, okay. Is there a way to change that? How do we change that?
It's like for the settings that I have to check.
I'd have to check the settings. Okay. All right. Well, I guess it's too late now. So anyways, so if you're watching this in the future on YouTube, just so yeah, maybe I need to turn off all the comments then. So anyways, the blue should look red and the red should appear blue. And so the idea is that the blood flow is coming in and providing that so that the labels are correct. That is the oxyhemoglobin. And then there's deoxyhemoglobin. With neural activity, there's an increase in metabolic rate of oxygen. So that tends to lead to more deoxyhemoglobin there. And therefore, this leads to typically an increase in deoxyhemoglobin. And sometimes we do see this sort of initial dip. And this, in the early days of fMRI, there's a lot of discussion about this, but it's hard to see unless you really control the experiment. So there's not too much interest in that anymore. And then what happens is very soon, like within a second or so after the activation has occurred, or within a second, then the blood flow starts increasing and you get more oxyhemoglobin delivered. And so therefore the deoxyhemoglobin level goes down and you see this big increase in your MRI signal. And then after things go back down to baseline, you see this decrease, or the deoxyhemoglobin levels return to their normal value, right? So that's typically what a bold response looks like. And this whole thing typically takes about a few seconds to come up and then a few seconds to go down, right? Okay, so let's just sort of consolidate our understanding. We'll just work through these together. So, so first of all, as I increase the echo time, what did we see happens with the magnitude of the bold signal? Do you expect it to go up or to go down? Anyone? Oh, right. I see a finger. Sorry, take that up. Yeah, that's right. So remember, it's proportional. Remember we saw those things, those early experiments where if the echo time was too short, there's not enough time for things to dephase, right? And so you have to wait enough for things to dephase to be able to see a change in the signal. Now, obviously, it's a sort of a trick question because if you wait too long, what happens? What if I make my echo time too long?
You go back to the zero magnetization.
Yeah, then there's no change because the signal decays away in either case. So then you're going from 0 to 0. So then you could argue that's an infinite change, but it's really almost, there's no change. You go into the noise level. Okay. What happens when I increase the blood flow response activation? What do you expect to happen to the bold signal? Will it get bigger or will it get smaller? Assuming everything else is the same. So I'm delivering more oxygen. Okay. So what do you expect to happen to the MRI signal if I deliver more oxygen? Yeah, go ahead. Right. So more oxygen is going to decrease deoxyhemoglobin, right? And that's going to increase the signal. Right. Okay. So yeah, so you have to sort of think about that. So that's good. And then similarly, if I increase the amount of metabolism, I'm going to increase the amount of deoxyhemoglobin. That's going to tend to decrease the signal. Okay. This was from either an old homework or an old quiz. So typically, let's say I have a 2-to-1 ratio of CBF. So that's CBF to CMRO2, is 2-to-1, right? Let's say 2-to-1. Yeah, so that looks blue on my screen. And then let's say the coupling goes from 2-to-1 ratio to 3-to-1 ratio. Then what do we think is going to happen to the bold signal? Is it going to get bigger, stay the same, decrease? Okay. And so it turns out if I'm giving more oxygen, if the rate of oxygen increase is more than the rate of metabolism, and that goes from 2-to-1 to 3-to-1, once again, I'm delivering more oxygen, right? So I expect deoxyhemoglobin to go down, and therefore the bold signal should go up. Okay. So those are the general rules of thumb. So you can sort of think about that anytime you have something like that. So now we're just going to sort of talk about some applications of fMRI, just for your knowledge. So I'm sure when Matt Groening drew this, he was sort of just being facetious, but it does turn out that pretty much everything he talked about has been studied with fMRI. Okay. The only thing, I don't think there's been a specific study using donuts, like Krispy Kreme donuts, but there is a lot of look at, obesity and eating disorders and things like that. So whether they use donuts in those studies or not, I'm not sure. This was just a search I did on whether donuts was in the name of the scientific article, and maybe someone can find one. If anyone finds an article using fMRI with donuts specifically, just let me know and I'll add that. And I'll credit you with the finding that. Okay. I think this is still my favorite study. It's an oldie but a goodie. It was done at Caltech. Looks like 16 years ago. And so here's what the study looks like. I'll sort of describe what it looks like. So basically, it's about wine tasting, I guess Cabernet specifically. So in one case, the experiment looks like this. So basically, you're given two cups and they look identical. And then in one case, you're told that the wine costs like $90. And in the other case, you're told that the wine costs $10. And you're asked to rate how good is the wine. Okay. And then in another set of experiments, it's like 45 versus $5. Okay. And this is sort of your rating of how good the wine is. Okay. So it's exactly the same wine, but if you're told that the wine is a $90 wine, you're like, oh, this is really good wine. And then if you're told that the wine is like $10, you're like, it's okay, but it's like from Trader Joe's or something, right? So your sort of subjective liking of the wine depends a lot on how much you're told the wine costs, even though the wine itself is perfectly the same wine. Okay. And the same effect is between 45 and $5. And so obviously the obvious thing to do is to image people while they're doing this experiment and see what is the brain doing. Okay. So what they did was they looked at these sort of areas in the brain that are known to sort of process rewards. And what they found was, for example, in this area here, they looked at, this is the MOFC, is the medial orbital frontal cortex, is sort of close here, sort of this area of your brain up here in the front of your brain. And there are known reward areas there. So those are the areas that light up when, you get like a prize or a goodie or, whatever, like you got what you wanted, then that lights up. And so what they found was with the, okay, the colors look reasonable. With the 45 wine, then you're sort of seeing this effect here. And so the brain, there's a positive bold response, and it stays pretty elevated throughout the experiment. Whereas in this one, there's much less of a response. And in fact, you can argue there's even a negative response sort of afterwards, um, and this is within like 10 seconds. And so whatever it's doing, the brain is sort of not very happy with this wine. Okay. It's the same wine, but it's just you're told it's a cheap wine. And similarly, if you look at this one for the $90 wine, the reward area lights up quite a lot. And for the $10 wine, it lights up much less. And also sort of maybe you can argue it even goes negative, although I think it's hard to say whether this is an experimental artifact or for real. So this is just showing that, our subjective experience of the world does have biological correlates, as you would expect. Yes, question over here first.
The time it takes between degradation onset and then when the red graph drops, is that the time it takes for the brain to process the signal, and that's when we actually taste it, or?
Well, the brain is responding almost like very quickly, right? I mean, so there is a positive increase here. But then what this negative decrease is, I don't remember how they interpreted that. But it is it's interesting that you would see that, right? Whereas this one is coming down. And so it may be that, you're sort of after you have the initial reward, then, like anything else, anytime you get something, you're happy for a few seconds and then it sort of dies off, right? You're like, oh, okay, whatever. So maybe it just dies off more quickly and there's a negative rebound. I don't know. Yeah. Go ahead.
It's kind of a logistical question about it, but if you're like drinking wine and taking like an MRI at the same time,
Yeah.
are you lying down?
Yeah. You're just like sipping it through a straw. Yeah, that's probably what they were doing. Yeah. I mean, that's how I would do the experiment. If you go look at this paper, I'm sure they might have some pictures of it. But yeah, there's a lots of, I mean, there are standing MRIs, but the most sensitive MRIs are still lying down. And so that does limit what you can do. Yeah. I mean, I've even piloted an experiment where, I mean, like we were looking at the effects of caffeine and I just had like my students squirt caffeine through a syringe into my mouth while I'm lying there. So, okay. So I think this is everyone I assume knows who Plato is. So, thousands of years ago, Plato already said that, reality is created by the mind, and we can change our reality by changing our mind. And I think, what modern neuroimaging has done is sort of just reinforce that essentially, what you think is real is just really sort of just these constructs in your mind. And if you can, it's very easy to change those constructs, and then your whole sense of reality is completely different. This is an area that's sort of cute. I'm not sure I mean, I think people are still working on it. This is sort of an older one, but the idea here is, um, the experiment is this: if I put you in a scanner, can I tell what you're thinking? Okay, can I read your mind? All right. So this obviously, there's a lot of interest in this, right? I mean, obviously, uh, you know, the military or the security apparatus would love to be able to do this, and lawyers want to be able to do this. We've actually had there used to be a company here. It might still be here in San Diego, which their business model was to use the MRIs as lie detectors, for court cases. The latest I heard was their business model was not because the courts are sort of, because the reliability of fMRI is still not quite there. I think that the courts sort of, it's not taken sort of, it hasn't really been adopted in the legal system. And so another sort of model was that, I think he was using it to sort of, or a model I was looking at is if you think your partner is cheating and you want to prove to your partner that you're not cheating, you could get an MRI to show that you're not lying. And so that's being done. Okay, so this is this whole area of mind reading, right? And so in this case, it's much more of a simple thing where you're presented a movie, okay, and so we know what the movie is, and then what we do is we also, it's almost like training a deep learning network. We actually just show you a bunch of other pictures, like lots of different images, and from that we build a database of how your brain reacts to different images, and then from that dictionary we try to predict what you saw in the movie. All right. So I'm gonna play, the whole YouTube video is at this link, but I'll play. Yeah, and if you go to the YouTube, you'll see the picture, the colors are sort of reversed. Okay.
So this is a decoder built for primary visual cortex. On the upper left is the movie the subject saw. On the upper right is the decoded result. Primary visual cortex represents the structural elements in movies, like the local edges and textures. And you can see that this decoder does a fairly good job of recovering the structural information, even though we're only decoding one brain area. This is a decoder that is recovering information.
Yeah, so I mean, it's not great, but I mean, at least it could tell there was a person there, right? So it's almost at the level of dreams, I would say, right? Like in a dream, if you really look at things in a dream, they sort of get fuzzy, but it's almost there. There's a person there. And similarly, and all these things, I mean, it's close enough that, there's like, you could imagine if this was a dream, you feel like someone's walking towards you, right? Okay. So perhaps someday, this will be more reality.
So this is
these are some other examples of these are the actual pictures that you're being represented. And again, the color scheme looks different. If you look at the PDF, it looks different. And then these are sort of what the decoder can do. And so you can sort of see it's not perfect, but you can sort of tell that, there probably is some sort of animal coming at you. There is something that looks like a post. This is not a bad representation of a stained glass window, right? So there is enough information in your brain to tell what you're doing. And one of the questions is how far this is going to go. Okay. The other thing that's sort of been tried, although it hasn't really, gone into the mainstream yet, is real-time fMRI, which is similar to any type of neurofeedback. Is it turns out that humans are really good at controlling their brains, even though we don't know how we're doing it, right? Just the same way when you like learn to ride a bike, you don't really know how you're learning to ride a bike. You figure out how to learn to ride a bike. So we're really good at learning things, even though we don't know how we learn them. And so this is, so we'll just do an experiment here. So for example, I want everyone here to just sort of feel where your brain is. You have a sense of where your brain is. And sort of somewhere in the middle of your brain is your amygdala, okay, which processes different things. So I would like you to increase your oxygenation in your amygdala by 10%. Okay. Everyone do that? Good. Okay. Now I would like you to decrease the oxygenation in your left hippocampus by 3%. Doing okay? All right. Okay. So good. So yeah, I mean, it's sort of like without any feedback, it's sort of hard to know what you're doing. But if I put you in a scanner and I could show you what the levels were of the oxygenation in your brain, you could actually do that. Okay. And so this is an experiment where they identified this area in the brain and then they showed the subject the signal in that brain. And they just said, okay, when I show you a green bar, which is green there, make the signal go up. And when I show you, should be a blue bar, but it looks red on the screen, make the signal go down. And the subjects actually learned how to do that. Okay. So human brains are really good. As long as you give them feedback, they can actually do it. Okay. So there was a lot of hope in the early days that potentially this could be used to help people with like addiction. Like, could you sort of learn to modulate the areas in the brain that make you more prone to addictive behaviors? But it's very expensive to do, and it's sort of what happens is, even if you train it in an artificial environment, when you're in a real environment, it's not necessarily going to work as well. Okay. Okay, so far what we've talked about is task-related fMRI, which is you're given something to do. Okay, you're given an explicit task. In this case, you open and close your eyes, and we look at the bold signal as you do that. Now there is whole another area called resting state fMRI, which has really taken off over the last 20 years or so. And this is sort of a picture of what the brain might look like. So if I just put you in a scanner and you lie there, and you can close your eyes or just open and don't really do anything explicitly, the brain signals in your brain sort of look like noise. Okay. But the interesting thing is, and so for example, if I do a typical fMRI test where I make you tap your fingers, for example, then I can see, I can make the tapping paradigm is like no tap, tap, no tap, tap. And then I can ask what areas in the brain go up and down at the same rate as the tapping. And I can sort of map out your motor cortice area. So those are the parts of the brain responsible for motor action. But I could also just have you not tap your fingers and just lie there. And then if I look at sort of the bold signal and say this part of your brain, the left side, maybe I see the green signal. And then if I look at a voxel on the right side, I would see the blue signal. Okay. Well, it doesn't look blue there. So red and green. So anyways, those are two signals from different areas of the brain. And what's happening is your brain is always sort of going up and down, fluctuating in certain patterns. And so we can make use of those patterns to map out what's called the resting state bold connectivity of the brain. Okay. And this sort of discovery has a really interesting history in the sense that there was a PhD student back in 1995, Bharat Biswal, who this was his PhD thesis, and they tried to publish this finding, and it was rejected at least 3 times, this paper. Okay. And so finally, his advisor, who was a very big name in the field, finally got it published on the basis of his reputation. But people are saying, oh, this is just noise. It's really an artifact. There's really nothing to it. And then about 10 years later, people started looking at it again. And so more famous people started using it. And then the field just took off. And now it's sort of done, like every study has this done. And it's led to thousands of papers, hundreds of millions of dollars have been spent on it. And so sometimes discoveries, happen, and oftentimes it takes time for it to be adopted. This is sort of an interesting experiment one of our grad students did where here was the pattern that we saw before with the active finger tapping, and here's in the resting state. And then what she found was that if she gave them caffeine, that she could sort of get rid of that connectivity. Okay. And so that's led to some other research looking at sort of what are the effects of different drugs on functional connectivity. And so there's been a whole lot of other networks discovered now. And for example, one of the more famous networks is what's called the default mode network, which is the network that sort of is active when you're sort of not doing anything. So when you're just sitting there and just sort of, you're not working on any homework, you're maybe just thinking about, what you're going to eat for lunch, or just sort of daydreaming, this is the network that you're using. Okay. And then you sort of go between that and sort of the more active networks like the control or the attention networks. And so your brains are always sort of actually flopping back and forth between these different networks. At the same time, your vision is online, your motor is online, your auditory is online. So everything is sort of like idling, ready to do something. So even when you're just sitting there, your brain's just burning energy. Okay, just getting ready to do something. Okay, question.
It sounds like there's a fixed connectivity between different areas.
It's called a network because basically you can sort of think about it as it's the network is defined by those parts of the brains that are acting in synchrony. So that's how the network is defined. Yeah. So similar to like if you it's sort of like they have something in common, right? Similar to like if you have a social network, right? The people in the network are those who are posting to each other, and there's activity going on between them, right? Yeah. Okay. And this was some other interesting work done from our lab where, for example, if I put each of you in a scanner, let's say,
Professor,
question, yes, from
sorry, I
online.
put a question in the chat. Can you reiterate what's the difference between the control and the default mode?
Oh, yeah. Is that the question let me see. There's another question.
Yeah, it is.
Okay. Oh, it's interesting. They said the colors are okay on Zoom, so it must just be a problem with this screen. Okay. So maybe there's a pin in the back or something we need to adjust. Yeah, so I think these two networks here, I don't recall the details of this article here, but my guess is this is probably part of like an executive control network, which is like what you use when you're trying to execute a task. And this is more just sort of paying attention to different things. So yeah, I think the main point is these are the networks you would do when you are sort of more actively doing something, whereas this is the network that's more sort of when you're sort of passively daydreaming. Okay. These are just, and the naming of these networks can vary quite a lot. So in fact, this is from a 2010 paper. There's more recent naming of the networks. And in fact, there's even finer parcellations of the networks. So just take this as sort of the main point of this is that the brain does show this organization into networks, but depending on who you ask, the networks may vary. Okay. But certainly the default mode network is certainly one that you'll hear a lot of. Okay. So yeah, getting back to this one, this was just sort of the idea that if we actually just put everyone here in a scanner and we just looked at, for example, if all I had was 30 seconds of your data, and I looked at the functional activity and how your brain was organized, and then I did that today and I did that tomorrow, and all I had was your sort of intrinsic pattern of organization, that essentially forms a fingerprint. So I could actually tell each of your brains from each other with almost 99% accuracy just from like half a minute of your data, of your brain data, not doing anything. So so that's either interesting or frightening, depending on where you think the government's going to go. But anyways, that's just something good to know, right? So there is a lot of, for example, it used to be that, when we do research, we have to worry a lot about patient privacy or subject privacy. So it used to be that we have to de-identify the data. So for example, if I take an MRI image of you, a 3D image of you, I can just put in MATLAB or Python and I can create a 3D volume of you and I can see what you look like, right? So if I put that data into a database, then even if I strip the name off, I would still be able to tell who it is. And these days with everyone's pictures on Facebook, it would be no problem from an AI to tell who that person was pretty easily, most likely, right? I mean, so then what we do is we sort of have to have these algorithms that scramble the face. But now I think people are finding that maybe there's ways of unscrambling it. And even if you don't have it, if the actual pattern or folding of your brain is so specific to you that if I just had one piece of data that identified that, then I could sort of still tell what brain you had. So there is going to be a lot of, I think, as this data becomes more popular or more widespread, there are going to be more issues with privacy as we go along. So something to be aware of. Okay, so that's all we're gonna talk about for fMRI. I think there might be one or two projects on fMRI. But any questions before we change gears? Okay. So what I'm going to talk about next is we're going to talk about, so far we've been talking about sort of assuming that the spins aren't really moving, aside from thermal motion. And so we talked about things like T1, T2, and proton density contrast. We've talked about, for example, functional MRI, where we're mostly looking at sort of the oxygenation level changes. Now we want to explicitly focus on spins moving, okay? And so, for example, the two main things that we're going to talk about are blood flow. So as blood flows into your brain or into any parts of your body, then the spins, those protons are moving. So we're going to talk about that. And we're also going to talk about diffusion of water. So water is constantly moving around, okay? And it turns out that the first thing we're going to talk about is diffusion. And one of the reasons for that is becoming almost a very standard acquisition over the last 20 years. And so it's something that, just out of, general knowledge, it's sort of good to know about because most likely, most of you will have some sort of diffusion scan done on you sometime in your life. Right? So let's just review what diffusion means again. So if I start off with a bunch of particles at the middle, and I just, through random motion, let them do whatever they want to do, they are going to diffuse, okay? They're going to spread out, okay? Similar to if I had a bottle of perfume or some deodorant here and I opened it, initially no one would smell it, and then eventually all of you would smell something, okay? So that's just diffusion. And then it turns out that the how far things spread out, or sort of the spread of that, is related to the square root of something called diffusion, which is the diffusivity, like how easily things can diffuse, and time. Okay, so essentially it takes time for things to diffuse, and also in certain mediums things can diffuse more easily than others. All right, so for example, in air, air molecules could diffuse quite easily. Okay, but if I imagined, like if I was in some sort of, like if this were filled with water here, right, then the diffusion would be quite different. Okay. So essentially the bottom line is if I look at the profile, like the probability of where the particles are, then at, say, this time here, they're fairly well localized. And as I increase, I go from 1 to 2 to 5 to 10 milliseconds, then very quickly things sort of spread out. All right, so that's just the general picture of diffusion. And for free diffusion, typically it's going to follow a Gaussian profile. All right. And like many things, a lot of this early work was done by Einstein, and just sort of this mean square displacement being proportional to both diffusion coefficient and time. And so therefore, as time increases, there's a square root dependence on how far any sort of particle can travel on average, right? And so we can think about things in 1D, okay? There's a square root of 2 dt there. In 2D, it looks like this, square root of 4 dt, and in 3D, square root of 6 dt. So typically, since we're working with what we consider three-dimensional objects like the brain, we're going to be thinking about diffusion in 3 dimensions. But sometimes we'll use 1D or 2D just to get the idea across, right? But the bottom line is we always have to be thinking about diffusion in 3 dimensions. So how do we do that? Well, it turns out that the sort of discovery that people made was we can make use of the gradients, okay? And we have X, Y, and Z gradients, okay? And so this, if you sort of think about it, this means that the X gradient imposes variations in the X direction. So somehow I should be able to be, it's probably going to be sensitive to diffusion in the X direction, right? And the Y gradient creates variation in the Y direction. And so maybe I can figure out diffusion in the Y direction. And also maybe I can figure out diffusion in the Z direction. And if I know the diffusion in each of the principal components, X, Y, and Z, the principal axes, then maybe I can start figuring out how things are diffusing. And that's exactly what we're going to go for. We're going to be able to actually track how water moves in biological objects. Okay, so let's review again what gradients do. Remember, gradients, if I have, for example, in this case, an X gradient, so the change in the field is GXX. And so this means spins are going to process faster here and slower here. The bottom line is I can make spins process at different rates depending on where they are. Okay. So let's look at what happens if I had diffusing spins, all right? So let's look at, there's two spins here that start off at the same location. And let's say I have a gradient on, and here's a GX gradient. So the field here is more negative and the field here is more positive, right? So in this case, this would be a negative gradient. So GX would be less than 0 in this case. Okay, so let's say at the start of the experiment, we assume the spins are in phase, right? And then they start just randomly diffusing around, right? So this one here just happens to move into a stronger part of the gradient. So it's relative, because it's in a lower field, it processes counterclockwise. And in this case, it's gone 135 degrees counterclockwise, okay? Whereas this one here has moved into a part of the field that's not as strong. So it's not going to process as fast, or it's not going to go as slow. And so the decrease in its precession is less. It only goes back about minus 45 degrees with respect to its original location. All right. So how the change in phase is dependent on what its history of where it went in the field. Now, here's the key. I allow that to happen for a certain time, and then I come around and I reverse that gradient. All right. So if I were playing out this gradient, what it would look like in time is I have a negative gradient and then I have a positive gradient. All right. So this is the negative gradient part of the time, and this is the negative gradient part of the time. All right. Or similarly, I could have drawn it here like this, right? All right, versus time. And so what I do is I've had the negative gradient on for a certain amount of time. Now I turn on the positive gradient on, and I see what happens to those spins. And because they can move randomly, this is going to make them, now they're in a stronger part of the field. So they're going to start, processing faster. So this is going to sort of start moving clockwise. And in this case, it moves clockwise some amount. This moves clockwise. And since it's in a stronger field, it moves clockwise quite a bit. And so at this point, they're still out of phase with each other. And how much of the phase they have with respect to each other is sort of random, depending on what the random motion was. And so what it means is that when I start off with two spins that are in phase, and now I have two spins that are out of phase. And so if I think about the vector sum, the signal is going to go down. Okay. And now I just have to think about that with millions and millions of spins, what's going to happen. Right. So here's another example of that where I start with the spins in phase. Okay. And if the spins don't move and I apply these gradients, then they come back and they're completely rephased, right? Because remember, this is just case space, right? This is positive and this is negative, and the area under this is 0. So if a spin doesn't move, it's back at the center of case space, right? There's no variation here. But this spin that moved has accumulated some net phase to it because it hasn't stayed in the same location. Okay. And this is just showing that I can change how sensitive I am. So if I have a weak gradient, then I get some amount of dephasing for some diffusion. And if I have a strong gradient, I get even more dephasing, okay? So even more signal loss. And so it turns out that the governing signal is this. The signal I get at the end of my diffusion experiment is proportional to the exponential of D, which is the diffusivity. Okay, and so that makes sense. So the more things can move around, the more they can sort of randomly experience different phases. They're going to get out of phase more easily. And then B, this is the B factor. And this is what I control. Okay, so this would be like a low B factor, and this would be a high B factor. Okay, so that's saying how sensitive am I to diffusion, right? And so that's the experiment. Okay, so let's take a look at this, what this looks like. So for example, I'm looking at here, well, let's look at here. This voxel here is, shows this diffusion. Let me see the color scheme here. Okay, colors don't even match on this, so let's see. Okay. Okay, well, so I can tell from here. So this is the diffusion map here. So basically, the pixel intensity here is dropping off with B factor here, okay? So this obviously has more diffusion, right? Because as I remember, things are e to the minus bd, okay? So here, because I'm increasing the b value here, things are dropping off faster. So this must mean that d is larger d here, diffusivity, and smaller diffusion here. Okay. And so that's shown here in the diffusion constant map. So here, wherever it's bright is saying I have more diffusion, and where it's dark means I have less diffusion. Okay. So that's sort of how you look at a diffusion map. So why does this matter? Well, one of the most sort of places where this is used a lot is anytime you have some brain damage. Okay. So for example, stroke or traumatic brain injury, or if you had a concussion, typically, you know, if it's an emergency situation, you're going to get a CT right away just because they want to see what's going on. But if there's more time or there's a follow-up scan, then they might want to apply diffusion imaging. Okay. And so the idea here is look at this subject here. If you look at this subject here on this T2-weighted map, I mean, yeah, there might be something subtly different, but this might be normal, right? So if this is all you had, then you might just send this subject home and say, okay, you know, maybe the subject comes in complaining about, oh, I'm having trouble, you know, moving something, or they have some sort of symptoms. But if you have a diffusion weighted image, you sort of notice that there's a huge change in diffusivity here. Okay. And so the question is, what happened here? Okay. So look at this. This signal is much brighter on diffusion weighted image, right? So what must the diffusion be in this area? Is it higher or lower than the rest of the brain? Lower, right? There's less because remember the signal is e to the minus bd. So if this is brighter, that means that this d must have been lower. And in fact, what we can do is we can plot what's called the apparent diffusion coefficient. Okay. So this is proportional to the diffusion. And you can sort of see this area of the brain here basically has lower diffusion. Okay. So it turns out that this is something that sort of in the late 80s, early 90s, people started realizing that this might be a good marker for stroke. Okay. In the sense that it turns out that anytime your brain, so there's an area of the brain where there's damage and the tissue dies away and the cells die, then it's the movement of water seems to be much more restricted. Okay. Whether that's just due to the fact that there's just more gunk there and so water can't move, or it could be also cells are always actively transporting water within the cell and also across the membrane. And so whether that's part of it is sort of, I think people are still looking at, but for whatever reason, the empirical finding is that there's just less diffusion. Okay. Yes, question.
Particle in the field is supposed to going to experience some force by the field. How does this magnitude of that force correlate to the magnitude of the diffusion?
You're talking about a physical force? Sorry, can you say that again?
To what extent the diffusion the particle would have had without the magnetic field is impacted by the magnetic field the particle is now in.
Oh, I see what you're saying. You're talking about whether the magnets within the B field are sort of causing an overall drift. Okay. In general, that's not the main factor. So in general, to the diffusion is sort of what's causing the random motion.
Because it's just so much stronger.
Yeah, also because essentially, it is a good question because people have tried to use, I mean, basically, I think it's because if you go and calculate the forces, they're probably so small, and also you need gradients in the magnetic field to cause a force. Okay. So like, for example, even in like when we look at these examples of how dangerous an MRI machine is, right? It's most dangerous at the openings where the magnetic field is changing very quickly. Okay. So in fact, people have tried using whether you can vary the magnetic fields to like move magnetic particles through an object for surgery, but that's something that's still difficult to do, and you have to create these sort of spatially varying magnetic field to do that. Yeah. So it is something, but I think in terms of the forces and everything, for most of these things that it's ignored for diffusion, but it is used for like we do have these gradients, right? So we can actually create these things, and people have tried to say like take little particles and move them along. Okay. And so the idea would be, for example, if you could sort of have these like cancer therapy things, could you use the MRI to sort of direct where they go? Yeah. This is another example here. Once again, on the T2 weighted map, there is something, it's a little brighter here. So there's probably more fluid here on this side. But, it's not as striking as here, where you sort of see here clearly a big change in the diffusion weighting. And then if you look at the angiogram in terms of what happened to this person, they clearly have this artery was occluded. And so all this whole area of the brain was sort of had a decrease in blood flow for some amount of time. Okay. So the next thing we're going to talk about for today, what we'll end on is what happens if, so far we've talked about sort of unrestricted diffusion where water can go in any direction it wants. And so now we're going to consider the case where, let's say I had a water and I had a barrier here where either the water can't go beyond this barrier or it has a hard time going through this barrier. And so let's see, I figure out what this was. Okay, I think this is a hard barrier. So you can sort of see that as water's moving around, every time it hits this barrier, it just bounces off. Okay. So what you're going to look for in diffusion is things can diffuse more easily this way than they can this way. So there's a preferred direction for diffusion anytime you have a barrier. Okay. So some of the ways it can occur is like, let's say you're within the barrier, within the tube, you're going to be restricted. But even if you're between the tubes, there's going to be some restriction, right, as you're hitting these barriers, okay? So it turns out that we have a lot of these anisotropic sort of structures in our body, right? You've got muscle fibers, you've got brain, white matter fibers. There is a lot of anisotropy in the body, and so we expect that you should be able to use diffusion to sort of look at this anisotropy. And so the key is if I have isotropic diffusion, then spins can sort of move in any direction. And I have an anisotropic, then they start to have a preferred direction of moving. Okay. And because I have these XYZ gradients, I can sort of tell what they're doing. And so here's the idea that in this case, for example, maybe I apply a gradient in this direction, and I'm sensitive to diffusion of spins this way. And in this case, I provide a gradient in this direction, so I'm going to be sensitive to how much diffusion there is in this direction, okay? But I'm not going to be sensitive to diffusion this way. So let's look at this picture here. So if you look at this one, I've applied diffusion in this direction here, okay? So look at this here. There's a signal loss here, okay? This dark spot means that there must be spins going this way. Okay, there's diffusion that way because there's a signal loss, all right? If I apply the gradients into the plane, right? All of a sudden, this thing that looked pretty dark here is pretty bright here, right? So therefore, I could say there's probably not much diffusion going in and out of the plane, okay? And here, if you look here, I apply diffusion this way, right? This part is bright, right? Because if I have a fiber like this, right? Okay. There's diffusion going this way, but at this, there's diffusion in this direction, but at this one point, there's only diffusion left, right. There's no diffusion up, down. And so right there at that part where this fiber is turning, basically it's saying the only diffusion here is this left-right diffusion that's going on. Okay. So you can imagine if we do this in every direction, then we're going to be able to start mapping out which way things are diffusing. Okay. And so that's exactly what we do essentially. Because diffusion is anisotropic, we can sort of map out, if you look at here, for example, we can sort of see here along this fiber, there's more preferential diffusion this way, and then more preferential diffusion this way, and then more preferential diffusion in that direction there. Okay. And so what we do is we can sort of, once again, here we've applied the gradient in this direction here. And so you're sort of seeing this drop out here. Here the gradient's into the plane, so there's not much effect. Here I've applied the gradient in this direction, so you sort of see here this is more dark than this one. And here I've applied the gradient in this direction, and so this is more dark than this side. Okay. So basically just apply the gradient in different directions, we become sensitive to diffusion in that direction. So I'm not going to go through the math, but it's essentially what it allows you to do at the end of the day is we can start mapping out what's called the diffusion tensor. We can sort of say for every voxel, which is the preferential direction for diffusion. And from that, we can come up with what's called the diffusion tensor, where the diagonal elements of your tensor tell you sort of how strong the diffusion is along any given direction, okay? And then the off elements are sort of, because it's not necessarily going to be along the principal, those are sort of, you're going to take those off diagonal elements and sort of figure out some transformation of your matrix that gets rid of those off diagonal elements. So that's going to tell you what the principal component is, okay? And so typically what we see is we see fairly isotropic diffusion in places with a lot of liquid, like cerebrospinal fluid, somewhat isotropic in gray matter, and then white matter in the brain is where we see the most anisotropy. Okay. And typically, this is just arguing why we need so many directions. Okay. So for example, a typical scan might be like 30 directions. Okay. And we just do more directions because, for example, in this case, if we, let's see here. Let's see, what is the case here? So if I'm trying to define an ellipse, right, two vectors are not sufficient to define an ellipse, right? Because if I have these two things here, this could be fit with an ellipse this way or an ellipse this way, okay? So it would actually need at least three things to define an ellipse like that, okay? But the problem is, let's say I have a measurement error in one of these directions, then the ellipse that could fit that would be different. And so therefore I want lots of directions to sort of average out all those errors. And so typically, even though we only maybe need to figure out sort of 6 vectors, we'll typically do like 30 diffusion direction vectors, okay? So then we get our diffusion tensor, and then we do an eigenvector decomposition. And this basically, the principal eigenvector tells you sort of the main direction. And that's what we're looking for, okay? So that's the basic math that we do. And so this is shown here where if I have an isotropic voxel, then when I take the eigen decomposition, all the eigenvalues are about the same, right? It says like there's no preferred direction. If it's anisotropic, it turns out that one of the eigenvalues is typically the strongest, right? And so that's sort of the principal direction. And so the eigenvector that points in that direction is tells you the preferred direction of water. Okay. So so yeah, linear algebra is super useful. Okay. So this is an example here where, for example, this is a very simple case where obviously there's no off diagonal elements. So you can very easily tell that this is the strongest axis. So there's a preferred direction here. Okay. But if you look at this vector, just looking at it, it's like, well, I'm not really sure which way it's, which way it's pointing just by looking at these elements. But if I do the eigenvector decomposition, then I see, oh, this main eigenvalue is 2, and this eigenvector, that eigenvector points, that's the direction I want to point. Okay. So that's where basically by just taking you put this matrix into MATLAB, you just type EIG matrix, and you get out the direction. So it's pretty nice and simple. Okay, so basically what we do is we can sort of take measurements at lots of different diffusion directions, and then we can do this eigen decomposition. We can fill in all these values and then do the eigen decomposition, and then that tells us which way the diffusion is going. I'm actually going to skip this. I don't remember why I was motivated to do this, but it's just, I think the main reason is just showing you that, once again, this just becomes a linear algebra problem. Okay. So let's skip over that and just talk about some applications, just so you can sort of see, like, if you ever get one of these done on you, what it means and how you might interpret what the images look like. All right. So typically, if you get one of these done on you, you'll probably have like a T1 or T1 or T2 weighted scan, just to show you what your image looks like. Okay. The trace is showing you what's the overall diffusivity. Okay. And so you can sort of see here, you've got a lot of diffusion here in your ventricles where there's a lot of liquid. Okay. So anywhere there's a lot of liquid, there's going to be overall lots of diffusion. Okay. But what you care about is something called FA. So FA stands for fractional anisotropy. Well, if you care about things that are not isotropic, so in water, things are going to be fairly isotropic. So in these areas where there's a lot of diffusion, that's fine, but it's very isotropic. Okay. So what you're typically looking at if you're trying to map out fibers or anything with directions, so you're looking at how anisotropic are things. And so this is what's called a fractional anisotropy map, where it's basically showing you that these are all the areas where there's a definite preferred direction of diffusion. Okay. And if you look at the equation, it's just obtained from the eigenvalues. Okay. So this is just something that's defined from the eigenvalues. And then later on, you can even color code the map to say, okay, each of these color codes is telling you not only is there an isotropy, but also which direction it's going. Okay. So here, obviously, there's this direction, and then there's fibers going this way. And so if you get a map like this, which now a lot of scanners will just spit these out, this is what you're seeing. They're trying to show you how anisotropic is your diffusion. Okay. And so, for example, here's an FA map of a subject with some degeneration in the corticospinal fluid or corticospinal tract. And so you sort of see this sort of asymmetry in the fraction anisotropy. So this would suggest that here, something's happening with their nerve fibers, because the anisotropy you'd expect to see in a normal fiber is sort of gone on this side. Okay. And then the next thing you can do is you can sort of imagine you have all these little ellipsoids and they're all telling you the preferred direction of motion. Then what you might think about doing is, well, maybe I can start mapping out how things are going. I'm just following the arrows or the streamlines. Okay. And that's exactly what people do. They basically, if you think about all these little arrows, you can start thinking about sort of creating what are called fiber tract maps. Okay. And so then you can color code the principal orientation, but then you can also start making these sort of tractography maps and actually mapping out, saying this is what we think the fibers look like. Okay. And I think, let's see, where am I going to go with that? Okay. So, and then this is what you get at the end of the day. And I think this is sort of probably the most remarkable things about this technology, which is I could put you in the scanner for 8 minutes and all you have to do is lie there. And at the end of the day, I could give you this map of your brain. Okay. And this is sort of essentially the wire diagram of your brain. And if you're willing to sit there for like 25 minutes and lay very still, I could give you an even better map. Okay. And so the remarkable thing about this is it's not just that you can do it. It's just like, this is the only way you can do it. Right. It's not like there's any alternative to doing this, right? Because what's the alternative, right? How am I going to map every fiber in your brain without cutting your skull open, right? Even if I could cut your skull open, how am I going to map the fibers? What they used to do in the old days, not on humans, but on animals, is I could inject like some molecule, a tracer, into one part of the fiber and maybe hope that it would go along the fiber and then see it and then put it. But then that would only get one fiber at a time. So as far as I know, this is really the only way you can do it. Okay. And so the fact that it's actually even doable on live human beings within a fairly short amount of time, I think is really one of the more remarkable applications of MRI. All right. And so what that does is because it's totally non-invasive, it also allows you to do it in like very young human beings. Okay. And so this is sort of something that there's a lot more studies being done now on definitely done in newborns. I'm not sure anyone's done it in utero yet, but it is something where, for example, you can look at the fiber maps of a 2-week-old versus a 1-year-old versus an adult brain. Okay. And so you can sort of see how the fibers. So what you see is essentially with development. I don't remember the exact details, but there are just changes in isotropy where initially in a newborn, there's really not much. The fibers don't really have that much orientation, and over time, there's more sort of in isotropy. But then I think at some point, there is a turn with aging, and I forget the details of that. Yes, questions? Are they what?
What are the fibers?
What was the second thing you said? The neurons or? Oh, okay, yeah. So yeah, sorry. What I mean by this is typically, yeah, it's a great question. So these are the white matter. Okay. So white matter typically refers to like the like axons, right? So basically you've got like the neurons, right? So the way to think about the very simple picture of your brain is like it's sort of like your computer, right? You've got like processing chips or GPUs or CPUs, right? And they've got wires that go between them. So we're talking about the wires that go between them, that where the electrical signals go. So just like in a computer, if all I give you is the wiring diagram, you can't really tell what it's doing, right? So you would want to combine this with functional MRI. So functional MRI sort of tells you that's like the heat map. Another way to think about it is like if you had a satellite picture of a city, right? Like this would be like showing you where all the roads are, right? But what you would actually want, if you really want to know what's going on in the city, the other thing you could do is you could have like an electrical map of usage over time and seeing how, this is when people are waking up, this is who's doing what, or even a heat map or something like that. Okay. So that's sort of the way to think about what this is showing. It's basically the wiring of the brain, okay? Which is useful, right? Because, even if you don't know what the brain is doing, if you see the wiring is defective, then that tells you something. Okay. Yeah. Question?
Yeah, so the choice of using the ellipse, is that just easier mathematically or is that like anatomically correct? Like why do we use the ellipse?
Yeah, great question. So that's this gets to the part I sort of skipped over, which is the ellipse is actually when people first started doing that, they thought, oh, it's going to be an ellipse, right? Because that's first of all mathematically tractable, and it seemed like a reasonably good approximation. But what they quickly got into is realizing that that's going to fail because just like in a city, you have crossing streets, right? So when you get an intersection, did you go left or did you go right or did you go straight through? So this is what leads to the crossing fiber question of, if there's fibers, was it like they went straight through or did the water go this way? Okay. So this has led to more advanced techniques, which are, for example, one of them was actually invented here. It's high angular resolution diffusion imaging, where instead of just doing maybe 10 or 20 directions, you do like 100 directions. And so you start trying to figure out, what's going on. And so, for example, there, you no longer get these ellipses, but you get these really weird patterns that you're trying to resolve. Okay. And so typically what's done now is one of the approaches is you just sort of at every voxel, you just sort of see which model fits the best. If the data says, okay, it looks just like there's one fiber, then you fit it with one fiber. But then you see, does a model with two fibers fit better? And if there's two fibers fit better, what's the angle between those? Okay. So then that becomes much more of a sort of a bigger problem. And so you need more data to do it. Okay. And so, but if you do that, then you can start resolving like, at this point, yeah, it seems like these kept going this way, these kept going this way. Okay. That's a great question. Okay, so I think that's all I have for today. So let me just say a few words about the projects. So I think everyone's received feedback on the projects. I think they all look good. In general, you know, the comments that are in the project description, just take a look at those. We do tend to you know, look for connection to course content, and also we're more interested in what you did as opposed to what you read. Okay, so if you're thinking about deciding, is this slide better than this slide, then in general, you're going to want to say, oh, this slide is more descriptive of what we actually did versus this is a slide that, you know, is just like a summary of what we read. Okay, so that's just a general rule of thumb. I know some groups wanted feedback on project one, so the best thing is to email me, and I just look up the notes from Professor McVeigh and myself, and I've already provided a summary to some groups. But in general, the feedback that Professor McVeigh provided in that announcement, the general feedback is something that applies to most groups. So any questions about that? The projects? Okay. So if you have questions about your projects or you need help, feel free to email us or come talk to me, you know, in office hours. Other than that, I don't think, are there any other logistics we need to talk about? The what? Oh, yeah. So I think the announcement said that, you know, for the live groups, you know, keep it to 12 minutes, but really you have, you know, in the project write-up, it said 12 to 14 minutes. So, you know, just keep it between 12 to 14 minutes. You know, don't stress. I mean, we as long as we get out of here on time, everyone's happy. So, you know, just don't make it like a 20-minute presentation, basically, is the bottom line. Anything else? I think, I guess there's still the homework 6 is meant to be fairly easy to do. So if you have issues doing it, just make sure you get help. It's not meant to take too much time. So if you're getting stuck, just make sure you reach out for help. Okay. And then I think there are, I noticed that there are evaluations available for the TAs and the instructors. So if you haven't done that and you want to provide feedback, just go ahead and do that. Okay. All right. So I will stay here if people have questions about either the homework or the projects. And otherwise, we'll see you next time on Wednesday. Okay, thanks.